Now we come to development of the spinal cord. Spinal cord develops from the caudal part of the elongated tube. Cranial part will form of the neural tube will enlarge to form the brain, and the caudal part of the neural tube is elongated to form the spinal cord. This neural tube increase in thickness by repeated mitosis of this epithelial lining, and it will form three layers from inner to outward, and the inner to outer layers are matrix zone, mantle zone and the marginal zone. The innermost layer which is lining the neural tube is neural tube or the neural cavity is called as ependymal lining or the matrix zone. This ependymal lining or the matrix zone, it undergoes repeated mitosis to form two types of cells. First type of the cells is neuroblast cells, which will develop into the neurons, and the second types of cells are spongioblast cells, and these spongioblast cells will convert into the neuroglial cells. Then we come to the second zone, that is mantle zone, which is present outer to the matrix zone. This mantle zone will further give rise to grey matter of the spinal cord, and then come the outermost zone, that is the marginal zone, and this marginal zone will form the future white matter. So the neuroblast cells, they migrate to the adjacent mantle zone. So initially neuroblast cells which are present in the they are formed in the matrix region, they will move to the adjacent mantle zone, and which will forming the spinal grey matter. And the axons of these neuroblast cells, they will going out into the marginal zone which is the outermost layer, and these axons of the neuroblast cells will form the future white matter. So thus the three layers, matrix, mantle and marginal zones are formed, inner to outer. Matrix zone will produce the neuroblast cells, and these neuroblast cells will move into the mantle zone and lead to they will form the mantle zone or the spinal grey matter. And the axons of these neuroblast cells, they are going out in the marginal zone, and these axons are myelinated and they will form the white matter of the spinal cord. Okay, so the mantle zone is forming the grey matter and the marginal zone is forming the white matter, and the matrix is forming the cells. These are neuroblast and the spongioblast cells, which will move into the mantle zone to form the grey matter. So cell bodies of the neuroblast will form the grey matter, and axons, which are axons of the neuroblast cells, which are moving out into the marginal zone, and they get myelinated and they will form the white matter. Now according to current theory, that neural tube, the wall of the neural tube consists of only one cell type, that is pluripotent neuroepithelial cells, and these neuroepithelial cells give rise to neuroblast. So these neuroepithelial cells which are giving rise to neuroblast cells, and these neuroblast cells form a covering around the neuroepithelial cells and called as mantle zone. So around the neuroepithelial cells, neuroblast cells are formed, and that are called as mantle zone, and it will form the grey matter of the spinal cord. And marginal layer, which is the outermost layer of the spinal cord, it contains the fibers which are emerging from the neuroblast cells in the mantle layer, and they are called as white matter of the spinal cord. So according to this theory, neural tube is lined by neuroepithelial cells which are giving rise to neuroblast cells. Neuroblast cells give rise to neuroblast cells forms the grey matter of the spinal cord, and their axons or the fibers emerging from the neuroblast cells will move into the marginal layer, and they form the white matter of the spinal cord. Now development of the functional columns. The cavity of the neural tube is like a slit, and due to formation of a vertical slit, dorsal and ventral wall of the neural tube is very thin. Here you can see in the diagram that the dorsal wall is called as roof plate and the ventral wall is called as floor plate, and the lateral wall is thick. And there is a sulcus in between these dorsal part. This is the dorsal part and it is the ventral part. And in between these two there is a sulcus that is called as sulcus limitans. And this sulcus limitans is dividing the neural tube into dorsal part and the ventral part. And the dorsal region, it is also called as alar lamina and it is functionally sensory. And the ventral part or the basal lamina, it is efferent or the motor. This is the basal lamina and this is the alar lamina. And from the dorsal root, here you can see the nerve dorsal root and this is the ventral root. Both join and they form the spinal nerve, which is the mixed nerve. Now coming to the development of functional columns. The cells of the basal lamina and alar lamina, they are arranged in longitudinal column. You can see 1, 2, 3 and 4. These cells are arranged in longitudinal columns. And these two afferent columns of alar lamina and two efferent columns of the basal lamina. So first two afferent columns of the alar lamina are GSA or the general somatic afferent. Yellow one is the general somatic afferent and the second one is the general visceral afferent. And the basal lamina, there are GVE and GVE and GSE or general visceral efferent and the general somatic efferent. So 4 cell columns in the spinal cord are termed as general because the 3 additional spatial columns, they are present in the brain stem. Here you can see the sulcus limitans, which is dividing the spinal cord into dorsal and the ventral part.